And for the last speaker in this session, I'd like to call up Jane Carlton, also from New York University School of Medicine, and she's going to give us the city view, ATMs, parasites, and wastewater.
Thank you. Actually, I'm not at NYU School of Medicine. I left about 5 years ago, but never mind. I'm at the downtown campus. Thank you. Thank you to Paula and to Lynn for inviting me here today. I've not been to one of these meetings before. I'm a parasitologist and an infectious disease expert. So I'm going to be telling you about my work my lab's work, looking at the parasites and the protists in New York City in particular. And actually I should say, for those of you who don't know New York City, this is an amazing picture of the city, an aerial view. This is Manhattan over here. NYU's about here. I think Paula lives about there, if that's not giving too much away. This is Brooklyn and Staten Island. Yes. Well, I'll have another map of it later on. Why do we study parasites or microbial eukaryotes or protists as they're known? Well, they're really fascinating organisms, and actually they comprise a really significant amount of the organismal and genetic complexity of our planet's biomes, and yet they've been very little studied in microbiome studies. And here you have a tree, small subunit ribosomal RNA tree schematic. It's a little old, but it shows you basically the 6 different lineages of protists. And it gives me the opportunity then basically to show you some of the really devastating diseases which are caused by parasites. So for example, here within the parabacillids, you have Trichomonas vaginalis, which causes the most common sexually transmitted infection, even more common than gonorrhea and syphilis combined. You also have here Giardia, which many of you may have heard about, beaver fever. In the Discoba, you have kinetoplastids, such as Leishmania and trypanosomes. In the alveolates, this is a very large lineage that has many parasites of important human health disease-causing importance. So here you have, for example, the malaria parasite and also Cryptosporidium 2 and Toxoplasma. Here you have plankton, diatoms, dinoflagellates, very important, entamoeba, and so on and so forth. And of course, here we are, animals. Actually, I can't find where we are because it's in blue. Green algae and land plants as well. So you can see a tremendous diversity then of these protists. And one of the lineages that we study in particular are these trichomonads, as I mentioned before. And I just wanted to show you this particular tree of trichomonads shown here, based on small subunit ribosomal RNA again, and some of the hosts, and how you can see that actually these particular species flip-flop between humans, cats and dogs, their pets, agricultural organisms, and wildlife as well. So in particular, Trichomonas vaginalis, which we sequenced the first genome sequence of about 10 years ago here, causes the most common sexually transmitted disease, but then Trichomonas stableri has been identified to cause this kind of particular cheesy mass in the mouths of certain species of bird, which is decimating those populations in California. And it's very closely related to Trichomonas vaginalis. In fact, there's probably been some species jump either from birds into humans or humans into birds at some stage. And then likewise Trichomonas tenax, which is found in both cats and dogs as well as humans. P. hominis, D. fragilis, which is associated with irritable bowel syndrome, but also found in pigs as well, and Trichomonas fetus. So very fascinating groups of protists. So why do we care about zoonotic parasites in New York City? Well, New York City itself is an amazing place, although I'm biased, obviously. 8.4 million people. Let's go back again. 300 square miles, so it's very population dense. It's a city of immigrants, and in particular here, this is a map taken from the New York Times a few years ago, where they said it's a montage of neighborhoods in which certain groups predominate. For example, a large Asian population in Chinatown indicated here. There's one pet for every household, so we love our pets in New York. And we have an enormous number of visitors, both international and national as well. So the sorts of questions we started to ask: what parasites and protists can be identified in New York City? Are there differences between the 5 boroughs, which you can see here, Bronx, Manhattan, Queens, Brooklyn, and Staten Island? How do the protists in one environment compare to the protists in another environment? And what are the sources and the sinks associated with parasites and protists? So first off, we optimized a method using 18S ribosomal RNA marker gene for amplicon sequencing. This, you can see here that we managed to identify a couple of regions, the V4 and the V9 region. So the V9 region had already been developed by the Earth Microbiome Project with a Illumina protocol, and it also has a vertebrate blocking primer as well, which comes in useful. And we compared it to a V4 region that we designed to as well, and we ran both of these on 23 parasite taxa and found that actually the V4 was much better for closely related taxa, V9 larger spectrum of taxa revealed, as you can see down here. We also curated the silver CHIME formatted database as well, added on our sequences, and we tested various lab methods such as bead PCR cleanup and automating the protocol as well. And finally, this was published a few years ago. So in this, we decided that actually using both the V4 and the V9 of the 18S ribosomal RNA unit was probably the best way forward, and having a depth of coverage at least 200,000 raw sequences to identify the protists. So we've used this particular marker and all of the methodology that we developed in several different projects that I'm not going to have time to talk about today. We were involved in the New York City subway project in terms of looking at the 18S component. Of course, that was published a few years ago. We also published a study actually looking into the methods for metagenomic sequencing of money circulating in New York City. This is one project I'm going to briefly touch about, and that's looking at both 16S and 18S ribosomal RNA markers on ATM buttons in New York City, and that was published just recently. And then I'm going to end up with talking a little bit about the sewage, the pests, and the pets project funded by the Sloan Foundation. So to talk about the ATM project, actually this was a project that was first started by Gloria, who we just heard about, and Holly Bick, who was a postdoc in my lab, pushed it forward to publication. And so we decided to take a study of ATM keypads in 3 different boroughs indicated here to describe the biodiversity and the biogeography using our 18S marker and then Gloria's 16S marker. We had a total of 66 samples from 8 different neighborhoods, and these had actually been divided into what are called NTAs. So we also had distinct demographic data associated with those areas, and some of them are indicated down here. So for example, in West Brighton, in Brooklyn, predominantly white population, European origin, and dominant ancestry was Russian. So some of our results, we found no clustering of 16S or 18S ribosomal RNA data sets due to geography or neighborhood demographics. You can see here, this is 16S and this is 18S analyzed by unify and a variety of different methods too. And in terms of the organisms that we found, shown in these panels here, so bacterial assemblages dominated by human skin communities, perhaps not so surprising, and eukaryotic assemblages dominated by fungal taxa and low diversity free-living protists, in particular toxoplasma and the parasite that my group works on, Trichomonads too. Some other interesting points, so microbial biomarkers that we were able to identify in these particular areas. Bony fish and mollusks enriched in Flushing and Chinatown. Chicken significantly enriched in central Harlem. And several fungal species as well that we identified. One which is utilized in industrial processes, the cause of black mold disease too. So interesting preliminary findings for this study. I think we concluded that actually keypads are amalgamate microbial assemblages from human, microbiome, eukaryotic food species, and extremophilic taxa as well, which are adapted to food, to air or surfaces in the built environment. And so we thought that this was a fairly good marker then for human behavior and environmental sources of microbes. But in fact, ultimately, and I should say this was published, as I mentioned. Ultimately, we really wanted to turn our attention to sewage because we hypothesized that in fact that New York City sewage is a much better biomarker for us really to look and identify for all of the protists in New York City. And one of the reasons for this is that New York City has a combined system, so residential and commercial waste, animal waste and groundwater runoff all end up in the sewers. In fact, there are 14 sewage treatment plants throughout New York City. You can see here they're labeled 1 through 14, also listed on the left-hand side here. And we set up a very strong relationship with the New York City Department of Environmental Protection, and we were able to sample each of these 14 plants over a year 4 times, so getting at least 14 samples 4 different times. And most of them were collected and given to us through this Newton Creek plant. These are the huge eggs which digest a lot of the sewage each day that is collected. So I'm just going to tell you some preliminary data because I'm worried about the time. So we did, as I said, quarterly sampling of 14 DEP sewage plants over 5 boroughs. I won't tell you too many details about that, but can sewage be frozen? No, so we had to extract on the day of. We thought a lot of the protist DNA would be swamped out by metazoan DNA, but the blocking primer actually helped us a lot here. And of course, we had this manually curated database as well. And just a couple of results here. So the first is New York City sewage is dominated by free-living protists. So here you have the V4 region, the V9 region, our different boroughs listed along here, and the organisms here. You can see that the most abundant taxa were things like freshwater ciliates in brown, kinetoplasts in sort of reddish brown, and cercozoans as well, indicated in yellow. And then we also found, of course, low levels of human-associated taxa in sewage, so V4 region, V9. Along here we have our boroughs, and you can see entamoeba, for example, species in blastocystis in those particular colors, and also listed here. And then also, once again, the Trichomonad species that we work on here. In particular, some very interesting large amounts of these Trichomonads indicated here. I'm not going to go into more detail about this particular study. PhD student Julia in the lab has a poster. Please go and see it. She'll be able to answer some of the questions that I put up here. But finally, what I did want to talk about is a study that we've, while we're analysing the sewage data, was to try and find out, well, where did the parasites and the protists in New York City sewage come from? And so we're undertaking a pilot study funded by the Sloan Foundation to determine the sources and the sinks here. And we've identified 5 different species that we're looking at: rats, dogs, cats, pigeons, and cockroaches. And we're collecting guts and feces, about 100 samples for each species, so 20 per borough, and then doing the sort of usual analysis here, 18S with the V4 and the V9, and then looking to predict sewage protist sources as well. And just to give you a quick update, we've actually, after a tremendous amount of work, collected close to 400 samples with a lot of help from vets, the public, neuter groups, and people, colleagues at Fordham University as well. So we have more than 100 species, 100 specimens of dog from the 5 boroughs, 80 of cat, 42 dissected midguts. So this is a picture of a dissected cockroach with the midgut that's been taken out here, done by Julia. And these were taken from the DEP, from many of the sewers where cockroaches live and then come out into the lower ground levels of houses and restaurants. And we also have a few pigeons too. So this work is ongoing at the moment, so I'm going to leave you hanging there. And while I wrap up, I did want to talk about, I ran a meeting a couple of years ago which was called Microbes in the City, Mapping the Urban Genome. It was written up in Nature here if you want to take a look at it. And it basically, one of my slides at the end, brought up these challenges that I thought were on the horizon for the community, in particular accessible representative databases, protocols, pipelines, functional validation of phenotypes. And then I also talked about how it's our duty as good scientific citizens not to scaremonger or to jump on the publicity bandwagon, to work with New York City or local officials to get permission to work in a particular public area. And that sometimes these aims can be in conflict with journalists who just want a good story. So I said ultimately it's our collective duty then to aim for generating robust data sets, undertake conservative data interpretation, and aim towards making New York City a greater place. Well, following on from that particular discussion, I should say that UniUke has now been developed. A consortium of microbial eukaryote specialists has come together to really pull together a lot of the standard marker, or develop standard marker genes and reference databases, and really come up with a solid universal taxonomy for microbial eukaryotes. So that's great news. And in addition, our own work has generated a lot of press, and this is something that's particularly interested me. We've been profiled for our sewage work in the New York Times and our pet and pest collection. As you can see here, this is when we did the Money Project, and this was a woman anchor holding up with a pair of pink tweezers, a $1 note here. And so this has been particularly interesting for me, so much so that we have come up with a one-day meeting that myself and New York City officials would like to hold. And here it is. It's called It's a Brave New World, the Promise, Applications and Regulations, Regulatory Considerations of Environmental Metagenomics in an Urban Setting. And we'll be hosting this in the summer of 2018. And really to bring together these disparate groups of people, scientists, especially next generation microbial ecologists, public health workers in the city who want to know how can they use the next generation sequencing in their day-to-day work, and then hopefully journalists as well, to see how the media can present microbiome and microbiology studies in an informative and non-misleading manner. Okay, so I would like to leave it there with acknowledgements, in particular Julia, who's been a fantastic member of the Carlton Lab and done an amazing amount of this work. Please go and see her poster. Colleagues, Gloria at NY School of Medicine, very many people who provided cat samples, as you can see here. Also the fish and wildlife samples from Krista Rogers, and actually a big thank you to New Yorkers as well for letting us sample. So thank you.
Yeah, so in San Diego, we do very little treatment of our sewage and just sort of dump it in a long pipe out into the ocean. But you have microbial digesters. Is all of it treated through the microbial digesters? And do you know, do you look at before and after the digestion to see if it's doing any of the...
No, we don't, because we want to find those parasites. We want to find those protists. And we're not, we don't want to find out how good a job the digesters are doing. I'm not entirely sure we will be allowed to have that data. So that's not what we're about, so we're looking beforehand. Yeah.
Right, because one of the advantages of the digester, of course, they produce, significant natural gas.
Right.
And they also could help sort of a water treatment. So if you did know that it was helpful in getting rid of this stuff, that could be useful for having to do less treatment later on if you put the digesters in the middle of it.
Sure, absolutely. And I think that's probably something that the city who run these particular digesters would like to see, can we use next generation sequencing for that. So the city has put their first MySeq into a public health laboratory, actually for the Legionnaires outbreak that occurred, so maybe that's something that they'll do further down the line.
Also, I had a question. You're using 18S? for most of your
18S, and actually some colleagues use 16S, and we've done some metagenomic shotgun sequencing too.
Yeah, that was my question. So the 18, do you get good results with shotgun metagenomics? Because it sort of depends on how many genomes you have available to match to. Do you get a
Right.
much better results with the 18S, or
Well, so it's, better. So there's not too much...
it's not better, but more data,
Yeah, lots more data. But the correlation between the two, we don't, 18S, you can get a completely different set of species compared to metagenomics. So I guess a combination of both is probably the way to go, maybe. I don't know. We're interested in the 18S and the Protists, so that's kind of good for me.
Thank you very much.